Hi, Miss Nikki here. We're going to talk about a section of Chapter 28 on pregnancy and human development. So the video right before this, you talked about the difference between a fetus and an embryo, and then I think the last thing that you ended up talking about were the 4 extra embryonic structures. So you talked about the allantois, or the what will be the umbilical cord. You talked about the amnion in its sac with amnion fluid inside. You talked about the yolk sac, and you talked about the chorion, or the outer structure there. So I just pulled this up to make sure that it's a good flow from the previous lecture. What we're going to talk about is actual embryonic development. How do we go from a gastrula to a fetus? You've already talked about implantation. She mentioned a little bit about the 3-layered embryos, but I'm going to cover a little bit more of it. So we started with that blastocyst, and that blastocyst is what implanted itself into the uterus, or the endometrium of the uterus. And then the blastocyst starts converting into something we call the gastrula. And I would do a little flow chart, since you have tons of new words to start with, right? You know, talk about the morula, talk about blastocyst, what does that turn into? That turns into the gastrula. So kind of keep a little flow chart there. So at about week 3 here, where I have it listed right here, you see an embryonic disc. And I'll have a picture coming up in just a second, where you'll actually see 3 separate layers, and they do a nice job in your book of color coding them, so you can tell the difference. And these are what we call primitive tissue. Sometimes they're also called germ cell layers. These 3 layers, every single body organ tissue, everything is going to be derived from one of these 3 layers. So these are your primary stem cells. So we have ectoderm, mesoderm, and endoderm. Ecto, outside; meso, middle; endo, inside, right? So the ectoderm is going to be your nervous system and all of your skin. We don't usually think of our nervous system as on the outside, but remember when we did dorsal, it is on the dorsal side. It is more towards the outer compared to your heart, your lungs, your liver, all that good stuff. Meso is going to be all other types of tissue. Again, your organs, your middle. And then your endoderm, remember how the inside lining of a lot of your organs have epithelial cells, right? Well, this is derived from endoderm. And there's a great, I hope I put it in here. Here we go. I'll go back to those in just a second. There's a great figure in your book. I want my students to know the 3 types of tissue and be able to give me an example of each one of those types of tissues. If I gave them a group or a multiple choice question, you could tell me which one is not part of ecto, meso, or endo. But this chart does a really good job of showing you the differences, and it's color coded and everything. So you can see the ectodermis, that's ectoderm, sorry, epidermis, hair, glands, brain, spinal cord. They talk about neural crest, but we're not going to really get into that. Endoderm, that's the internal lining, or the epithelial lining. When we talked about blood vessels, we used the word endothelial, right? So that internal lining in your blood vessels, that would be endoderm. And then mesoderm is really everything else. I mean, you can look at this list here. Myotome, we're talking about muscles, and remember, myo means muscles. Kidneys, gonads, connective tissue, bones, joints, ligaments, walls of your respiratory tract, your heart, your blood vessels. So mesoderm is really the majority here. Okay, let's go back and just look at the picture. I have it highlighted here in red for you. So when that blastocyst implants itself, when we start gastrulation or turning into a gastrula, you'll definitely see these 3 separate layers. This disc develops internally. And you can see that the red here are meso, the yellow is endo, and the blue is ecto. So these are primary stem cells that are going to be differentiated or told what to do. So at some point, there's going to be chemical signals that say, you're going to become heart cells, you're going to become blood vessels, you're going to become bones. So in the very beginning, they're all the same, but eventually they will be given a specific job, and we call that differentiation. Just something to bring up here, why are people interested in using these cells for stem cell therapy? Why do they want to harvest an embryo at this particular point in time? So you can think about that for a minute. I just said they're stem cells, right? If we can find a way to program them, we can have them turn into any cell that we want. So let's say that we pull some mesoderm cells out of here. We can tell some of them to become heart cells, and some of them become lung cells, and some of them become liver cells. So this is the ultimate way to create tissue that you want. It has not been programmed yet. It has all of the capabilities to become a multitude of different cells. Okay, so after gastrulation sets in, we're going to have organogenesis, and that tells you basically what it is. You're generating organs, right? What I think is interesting is by the 8th week, so by the 8th week, almost all organ systems are recognizable, and we call this the end of the embryonic period. We are now moving into the fetal period. So that's the definitive line there. At 8 weeks or 2 months, organ systems are developed. No longer an embryo, we're now going to call it a fetus. So the fetal period is much longer, but you can see that a lot of stuff happened in the embryonic phase, right? All right, so fetal development 9 through 38 weeks. This is rapid growth of body structures. The initial body plan is already laid out. So what's going to happen in the fetal period? Everybody's just going to become fully functional. I'd like you to look through this table. I just think it's interesting to kind of see the changes that are going to occur in the fetus from 8 weeks to 9 to 12 weeks. And there are different things that we've talked about in the past that you can see. Ossification begins about week 8. You're going to have some muscle contraction. Cardiovascular system is fully functional at 8 weeks. Heart has been pumping blood since the 4th week. Obviously, you have the head is quite large. It's not really in proportion to the body. We move into the 9 to 12 weeks. You can see that the sex is kind of the genitalia, external genitalia, the sex of the child is becoming a little bit more easily detectable. Blood cell formation begins in the bone marrow. Skin and epidermis is obvious now. As we move to 13 to 16 weeks, some of your more organized neurons, like in your cerebellum, which control your coordination, those are going to start developing and becoming prominent. Glands are going to develop in your gastrointestinal tract. Your kidneys are going to kind of start developing their structure and function. We now have joint cavities present. And then at the 20 to 30 week mark, you're going to see myelination of the spinal cord. Hopefully remember myelination, the protein lipid that's wrapped around that helps with conduction of signal. So you can kind of look through this table and just kind of get an idea of at about what time would certain organs be functioning. Okay, so now we'll move into labor. So what's going to happen? So during the last few weeks of pregnancy, I have a whole bunch of things listed here, and then we have a picture coming up, and the picture will help a little bit with some of these. Fetal secretion of cortisol. Remember, this is the stress hormone, is what we called it. So the fetal, released into fetal circulation is going to be cortisol. This cortisol is going to stimulate the placenta to start excreting estrogen. Estrogen increase is going to cause the production of oxytocin receptors. Hopefully we remember oxytocin back from the endocrine system. So oxytocin is responsible for uterine contractions, right? So if you want to have a bigger bang for your buck, so to speak, you need to upregulate your oxytocin receptors. So what does this mean? This means the uterus is actually triggered to make more oxytocin receptors. So when the oxytocin is released from the pituitary, that it has more binding sites, therefore you have a bigger effect or larger contraction. Another thing that's interesting, we'll come back and talk about this when we get to respiratory, surfactant. Surfactant is a protein, and it has to do with your lungs. When the fetal lungs are developed enough, this surfactant is going to be secreted. And this surfactant also helps, you're going to see in just a second that the cervix has to efface or open large enough for the head of the fetus to get through the vaginal canal, and that this surfactant actually helps soften the cervix. But it's also something that we look at to tell whether the fetus is fully developed. So we look at the surfactant levels, and surfactant has to do with keeping the surface of the alveoli in your lungs open so that you can exchange gas. And that sounds a little technical, but we'll get to it in respiratory. Also, there's fetal oxytocin released. Remember, the mom has a pituitary. Well, this fetus is completely developed. It has a pituitary as well. So the fetus is going to also release oxytocin, and that's going to also induce the placenta to produce prostaglandins. So remember, prostaglandins are going to help with muscle contraction. That's what they're known to do. So this statement at the bottom, both oxytocin and the prostaglandins are going to stimulate very powerful uterine contractions. So this is the picture from your book. You can see the estrogen from the placenta. That's going to cause more oxytocin receptors to be expressed on the uterus. Oxytocin from both the fetus and the mom's posterior pituitary are going to stimulate the uterus to contract. The placenta is also going to secrete prostaglandins. That's going to cause more vigorous contractions, and those are both positive feedbacks. Remember, the larger the contraction, the baby's head hits the cervix. The baby's head hitting the cervix sends a message to the posterior pituitary to release oxytocin. Oxytocin is released, furthers the contraction. Baby's head hits the cervix, causes this entire circular loop. That's what the positive feedback is. Remember, you're driving towards one ultimate goal, and that's expulsion of the fetus from the vaginal canal. So let's just go through the stages of labor. I put in just a few things here. I don't think you'd have to know everything about the different stages. In your book, they show you a picture of both early and late dilation. I just put the one picture up here, but if you want to go look at the early, the cervix is still intact. And let me show you where that is, because it's a little bit hard to see in this picture. No, that's not what I want. That one. Okay, so this is what's left of the cervix. So we know that usually the cervix is a very, very tight, narrowing passage between the vagina and the uterus. Here, this is the word effaced. It means obliterated or to be gotten rid of. And you can see that it once was the cervix that was real tight, and now it is pretty much opened up and flattened against the wall of the vagina. So that's what effaced means. This is usually the longest. It says anywhere between 6 and 12 hours. You're going to start with very weak contractions. They're going to become more vigorous and more rapid and last for longer. The cervix is going to efface fully. When it fully dilates, it's about 10 centimeters. So you'll often hear when people are in labor, oh, she's at a 2, oh, she's at a 3, she's at a 7. They're talking about the cervix and how far it has effaced. And you can see that this will be a problem if it doesn't. So if the cervix stayed narrowed and together and did not efface, what's going to happen? The baby's not going to be able to get out of the vaginal canal. Eventually what's going to happen is the amnion is going to rupture. Remember, the amnion is the membrane that wraps around the fetus, and there's embryonic fluid on the inside. Well, eventually during all this contraction, that amnion is going to rupture, the amniotic fluid is going to leak out. So we've all seen the movies where the person's standing there and it's like, psh, and there's water all over the floor. It's usually not that much water, but it is enough to make a mess. So that's what's happening during that part. The amniotic membrane is rupturing and the fluid's being released. And then the last thing, they call it engagement. This is when the head is actually entering the true pelvis. So the baby's head is actually on its way. All right, so let's move on to expulsion stage. During the expulsion stage, you're going to have really strong contractions about every 2 to 3 minutes, lasting for about a minute long, and this is where you're pushing. When they tell you not to push in the beginning because you could be pushing the baby's head against the cervix that's not fully effaced. When you get to this expulsion stage, the cervix is fully effaced, you have really strong contractions, you have a clear pathway, and you can push the baby out. So this is where we have the delivery of the infant, and you've seen the word has changed, right? It's not fetus anymore, it's infant. The last stage, remember we developed this intricate system, this placental system where we have chorionic villi, and we have pretty much capillary exchange between mom and baby, and so all of this has to be removed. It is pretty much, I don't want to say stuck to, but lining the uterus. This has to come out. So after the baby during the expulsion stage, after you get rid of the baby, and that sounded bad, you know what I meant, the placental stage, they'll say push one more time. And when you're pushing, you're actually pushing out what we usually call the afterbirth, but this is actually called the placental stage. So the placenta is becoming detached from the uterus, and one strong push, and usually that placenta and umbilical cord and everything comes out with one last push. This slide here is just talking about how the infant has to adjust to being in an external environment, right? It was in this nice warm cocoon. It got food. It got oxygen. It didn't have to really do anything but grow. It was suspended in this nice buoyant fluid, and now it's been pushed out into the world, and we call this the neonatal period. It's a 4-week period immediately after birth, and all of the systems, the infant's system functions have to start working, right, on their own. They're not getting mom's food and mom's blood anymore, right? So initially, that very first 1 to 5 minutes after they're born, they usually do something called the APGAR score. So you get 1 to 2 points, depending on different factors, for all of these things that follow. How well is the heart beating? How's respiration? What color is the baby? And a color has to do with whether the liver is really working or not, right? Muscle tone, reflexes. They do, I believe it's called the Babinski's reflex test, where they run their finger up on the bottom of the baby's foot to see, what does a reflex tell you? A reflex is going to tell you how the central nervous system is working, right? And then they give it a score for up to 2 points for each of those 5 things. So anywhere between 8 and 10, healthy baby. So you usually see APGAR score, you'll see a number. But when they take the baby away right away after childbirth, that's what they're doing. They're assessing that APGAR score. And then this last statement, I'm mentioning it now because it will make sense when we get into respiratory system. But what's happening at first is the baby's not receiving mom's oxygen anymore, right? The baby is expected to breathe on its own. Its lungs should be fully developed and working. So an increase in carbon dioxide in the infant. The baby doesn't know to breathe yet, right? So carbon dioxide builds up in the baby's bloodstream. This triggers something called central acidosis. Too much carbon dioxide makes the blood acidic. And we know we don't want our blood to be acidic, right? We want it to be 7.35 to 7.45, right? Having very acidic blood or having too much carbon dioxide in your blood triggers the respiratory control centers in your brain to trigger the first inspiration or breath. And we'll see more of this when we talk about respiratory. If your carbon dioxide levels are building, right now as I'm standing here, if my carbon dioxide levels are building up too much, my respiration is going to try to accommodate. So if I have too much carbon dioxide, I'm going to start breathing more, right? Trying to get the carbon dioxide out and try to get more oxygen in. And that's exactly what triggers the baby to take its first breath. And they're usually really angry about it, right? Probably would be too. There's also a transitional period. The first couple of hours after birth, they're being monitored. The baby's going to wake up every 3 to 4 hours. They're going to be hungry. Pretty much they're adjusting to the different environment. The last thing I want to talk about, it's a little bit more difficult concept. We got to go back and kind of revisit the heart and the blood vessels. The picture in your book is kind of iffy. It's okay, I'm going to use it, but there are some really good animations out there where you can actually trace the blood flow, like little arrows are moving so that you can see fetal circulation. I usually just make my students understand the difference between the two and some of these things called vascular shunts. But you'll have to ask your instructor. Some may want more information from you. So basically what we're saying is when the fetus is developing, it has a unique circulatory system. It's not the same as it is at birth. There are 3 vascular shunts, and I'll explain this when we move to the actual figure from your book. Remember, I just said that the mom is giving the baby blood, right? And the mom is getting rid of the baby's waste. So there are some shunts or offshoots in the fetal circulation that occlude or close normally after birth. They're only there during the fetal period. Once they're an infant, there's triggers before birth. Okay, so here's the figure from your book, the side-by-side, the fetus and the newborn. And let me get the pen working. No, come on. Okay. So right here, we're going to see this is the placenta, right? This is the umbilical cord, and it's delivering oxygenated blood. So highly oxygenated blood is in red here. So the first bypass, or the first shunt, happens at the liver. So you can see right here, some oxygenated blood is going to the liver, but normally, go to the newborn side, normally in us, what's happening? We have inferior vena cava, we go off, branch off into the liver, well, I should say inferior vena cava is here, this is your hepatic portal. So you're delivering the deoxygenated blood through your liver, and hopefully your liver is filtering out any toxins before it gets to the inferior vena cava and dumps it into your atrium, right? We don't want to get anything back to the heart that, you know, any bacteria, any toxins. So your liver is supposed to filter that out before it gets to the heart. But what about the fetus? Their liver isn't fully functioning yet, and the mom has hopefully already gotten rid of some of the toxins. Again, this is why you have to be careful as a mom on the food and things that you put into your body when you're pregnant. But in this particular instance, the oxygenated blood is just going to be delivered to the liver and help the liver grow. The rest is going to, oxygenated blood is going to go to the inferior vena cava, and it's going to mix with the blood, the deoxygenated blood that's circulating the fetus. So that's why here they have it in purple, because they're trying to show you that it's an actual mix. Pretty much you have deoxygenated blood coming in and highly oxygenated blood, so you get kind of this moderate oxygenated blood. And then what's going to happen is the blood's going to go here into the right atrium, and then there, right here, there's the foramen ovale, and this is another shunt. It is a bypass of the pulmonary circuit. There's also a ductus arteriosus, and that's another bypass of the pulmonary circuit. Again, are their lungs fully functioning right now? No, they're not breathing in oxygen, they're not getting rid of waste, mom's doing that for them. So there's no reason to go out to the pulmonary circuit to exchange gas, right? So this foramen ovale actually allows for blood to go directly here into the left ventricle and go up and out the aorta. So this is moderately oxygenated blood that's going to go out the aortic arch, and it's going to go out into the fetal circulation. It's low oxygenated, but it's still oxygenated blood, right? And then eventually the waste is going to come back out through the placenta, be exchanged with mom, and she'll get rid of the waste. So 2 bypasses for the pulmonary circuit, 1 bypass for the liver. Hopefully, when the infant is born, these bypasses have been closed. And what they do is they show you as a fetus what's it called, and then as a newborn, what does it turn into. So you can see here, here's the little ligament that used to be the ductus arteriosus. This foramen ovale will close and will not let blood flow through to the other ventricle. And I said normally on the slide before because sometimes there are problems. A child could be born where their foramen ovale has not completely occluded or closed. So what's going to happen now? They're going to have blood flowing into their left ventricle that's supposed to be oxygenated, right? This is supposed to be oxygenated going out to the body, where they're going to have deoxygenated blood flowing into their left ventricle. So some problems do exist, but just look at this picture, see if this kind of makes sense to you. Understand, why do we have to bypass the pulmonary circuit as a fetus, and why do we have to bypass the liver as a fetus? Again, if you want to Google, there's some, I don't have a website for you, but there's some good fetal circulation, actual animations where you can see how the blood is actually traveling. I haven't looked at my A&P lately, and I can't remember if they have one on there or not, but I would look at that as well. All right, well, I think that's it for my lecture today. Hopefully that made a little bit of sense, and I'll see you next lecture. Bye.